Hello everyone, I am Mohammed Mehrab from the Department of Biochemistry and Biotechnology, USDC. Today I will briefly present allele-specific PCR or ASPCR, which is a PCR-based technique used to detect specific genetic variants or mutations in DNA. Before understanding ASPCR, we need to know what an allele is. An allele is an alternative form of the same gene. Different alleles arise due to change in the DNA sequence. Some alleles are normal, known as wild-type alleles, while others contain mutations. ASPCR helps identify which allele is present in a DNA sample. Here is a figure which compares a normal allele and a mutant allele. Let's talk about the principle of allele-specific PCR. ASPCR use primers designed for a specific allele. The 3' end of the primer must perfectly match the target DNA, and DNA polymerase can efficiently extend only a perfectly matched primer. If a mismatch occurs at the 3' end, amplification is greatly reduced or it may be absent. Let's look at how the experiment is performed. The first step is DNA isolation from the sample. Next, allele-specific primers are designed, one for the normal allele and another for the mutant allele. Finally, the PCR reaction mixture is prepared using template DNA, primers, DNTPS, DNA polymerase, and PCR buffer. Once the reaction is prepared, PCR amplification begins. It involves three steps: denaturation, annealing, and extension. Amplification occurs only when the primer perfectly matches the target DNA sequence. After amplification, the PCR products are analyzed using agarose gel electrophoresis. A band with the normal primer indicates the normal allele. A band with the mutant primer indicates the mutant allele. And bands in both reactions indicate a heterozygous genotype. The gel image here demonstrates different band patterns and how they are used to identify the genotype of the sample. So, in summary, ASPCR is a simple and effective technique for detecting specific genetic variants through allele-specific primer binding. That was all about allele-specific PCR. Thank you for your attention.